The first problem we will look at is 0.15 molar NaCl solution is isoosmotic and isotonic. If a cell were to be placed in the solution, what would happen to cell volume? The first bit of information that we need to know in order to solve this problem is that inside animal cells there's a 0.3 molar concentration of solutes. And so we'll use that to compare it to the 0.15 molar solution of sodium chloride. We know that sodium chloride dissociates into both sodium and chloride, and so this actually is like saying 0.15 moles of sodium and 0.15 moles of chloride. And so if we add those together, then we have a 0.3 molar solution, pretty much. And so if we're comparing these together, then it would be isoosmotic because they are the same concentrations. We know that NaCl is a non-penetrating solute, so it will affect tonicity. It will not cross the membrane of the cell, so it is isotonic. The solution is isotonic. So if we draw the cell here, that means that although water can pass through the membrane, it is coming in and coming out at the same rate, and so the cell will remain the same size, therefore isotonic. The second question we're going to answer is 0.3 molar NaCl is blank osmotic and blank tonic. What would happen to cell volume? Again, to answer this question, we'll start by stating that there's a 0.3 molar concentration inside the cell. Comparing that to the 0.3 molar concentration of NaCl, remember that's like saying 0.3 molar concentration sodium, 0.3 molar concentration chloride. Together they equal 0.6 molar concentration outside of the cell. That is greater than the 0.3 inside the cell, so it's hyperosmotic solution. Remember that solvent follows solutes. So because there's more solutes on the outside of the cell, the water from the inside of the cell will move out, which means the cell will shrink, so it's a hypertonic solution. The next two questions have to do with urea instead of NaCl. So this first one, we have a 0.15 molar solution of urea, and that is blank osmotic and blank tonic. And what would happen to cell volume if a cell was placed inside the solution? Alright, so we have a 0.3 molar concentration inside the cell compared to the 0.15 molar concentration of urea, and it doesn't dissociate, so that won't be a problem. But remember that it is penetrating. That won't really affect anything in this problem though. Because the concentration inside of the cell is greater than the concentration outside of the cell, it is a hypoosmotic solution. And I write it wrong here on the paper. I say hypotonic, but I meant hypoosmotic. We'll talk about tonicity afterwards. I'll fix that later. So if we draw the cell, the water would be going into the cell, and the cell would expand because there's a greater concentration inside the cell than outside the cell, so the water outside would follow the solutes to the inside. A 0.6 molar solution of urea is blank osmotic and blank tonic. If a cell were to be placed in the solution, what would happen to cell volume? So we'll compare the 0.3 molar solution inside the cell to the 0.6 molar concentration outside of the cell of urea. The 0.6 molar concentration is greater than the 0.3 molar concentration inside the cell, therefore the solution is hyperosmotic. In the last problem we did, the fact that urea can cross the membrane didn't affect the solution when we talked about tonicity, but because 0.6 molar is so much greater than 0.3 molar, when a cell is placed in this solution, both the urea particles and the water will flood into the cell, causing the cell to swell. Because the cell swells in these conditions or expands, this is a hypotonic solution. So notice that this solution is hyperosmotic, but hypotonic. So this question was addressed on the last video, but on this video we will work it out by hand. In this question, we'll have a tub of solution, 15 liters on one side and 30 liters on the other side of a semi-permeable membrane of a 0.9% NaCl solution. And the question asks, if you add 1 liter of water, how much of the liter would you need to add to side A and how much would you need to add to side B if we wanted to maintain the equal osmolarities? To start solving this question, we will add the liters together, so 15 liters and 30 liters for a total of 45 liters. Then we'll take the 15 liters on side A and divide it by the 45 total liters and then multiply it by the 1 liter of water to get 0.33 liters that we will add to side A, so about a third. And then we'll do the same thing with the 30 liters on side B. So 30 divided by 45 times 1 liter, and so that's about 2/3 of the liter to side B. So when we add the 1/3 of the liter to side A and the 2/3 of the liter to side B, that makes it so that there's an overall volume increase in both places, but it stays as a 0.9% sodium chloride solution on both sides as well. So the last question that we'll work through on this video is, if you drink 2 liters of water, how would the volume and osmolarity of the ECF and ICF change? Here's the reference table that we'll use for this problem that you can come back to and review as we work through it. To begin, we're going to figure out the new total body volume. So we take the 42 old liters and add the 2 new liters to it for a total of 44 new liters of the total body volume. Then we'll figure out the new ECF volume by taking the 5100 osmoles and dividing it by the total 12,600 osmoles. So that's adding the ECF and the ICF together to get that total 12,600 osmoles. And then we'll multiply it by the total 44 liters of body fluid, and then we'll get 17.8 liters of ECF for the new volume. The ICF volume will be the original 7,500 osmoles divided by the 12,600 total osmoles, and multiply that by the total 44 liters to get 26.2 liters. Then the new osmolarity, we'll take the total osmolarity, I guess it's in milliosmoles, all of these, but the other ones cancel out, so it's fine. And then we'll divide it by the 44 liters to get 286 milliosmoles per liter.